In this video protocol, we demonstrate in detail a culture system for growing bovine oocytes. This system supports the oocytes viability in culture for up to 5 days and allows for their differentiation. Using this culture system, oocytes collected from small antral follicles, which are normally not used in standard protocols for in vitro embryo production, can be grown to increase the availability of fertilizable gametes.
This exploitation of the ovarian reserve can provide new options for the genetic salvage of threatened species of the bovine family or of local breeds at risk of genetic erosion.
To begin, prepare 15 milliliters of basic culture medium and supplement it according to manuscript directions. Next, prepare 3 milliliters of holding medium by adding 5 micromolar silostomide to the basic culture medium and pour it in a 35 millimeter petri dish. Prepare long IVCO medium by supplementing basic culture medium according to manuscript directions. Add 200 microliters of long IVCO medium to each well of a 96 well coated plate. Then fill the 4 edges with sterile water to compensate for evaporation and to maintain appropriate humidity during culture. Incubate the 96 well plate and the holding medium at 38.5 degrees Celsius and 5% carbon dioxide with maximum humidity. Wash the ovaries 4 times in sterile saline maintained at 26 degrees Celsius. Then aspirate all follicles that are more than 2 mm in diameter with an 18-gauge needle connected to an aspirate.